So we'll skip that one. Because you knew right away that, let's see if we can get 100 on this thing, that identical sibling was the one where it matched up perfectly. Sibling or parent was the one that was a little off, but mostly right. And then unrelated was this one. Next. Cladogram. Did it look familiar? Was this one on your test? I put two of them on different versions of the last two tests. So basically, the organisms are the letters. The differences in amino acids, the count of that differences is an intersection, and they've given you D. It's the only thing that's on that cladogram is organism D. So what are the two most closely related organisms in this whole deal? B and C, because the intersection of B and C shows there's only one amino acid difference. And so that tells you that those are B and C are either going to go on this fork or this fork, and it's not this one because D's already there. So you know B and C have to go here and here. So B and C, and then it's just a matter of deciding where. Oh, and notice it doesn't matter whether B or C is on top, right? It's going to be right either way. There's some wiggle room in those drag and drop ones that it's helpful to know about. So the question is A or E more related to D? And so we had to kind of go and find, all right, A and D have 11 differences. D and E have only 3 differences, right? So where does D go? No, where does E go? Where does E go? With D, on the fork with D. So no problem there. Which means A is left over here. Any questions about that one? And if you're wondering why there's so many cladograms on these practice tests, it's because there's been a lot of cladograms on the last like 3 end of course exams. So there's a reason I had you go through all 6 of those missions in the Nova Labs thing. Next is number 3, the other cladogram that I put on tests. This one looks real scary at first until you figure out what 2 things. What's one thing that really helps on this one?
It shows you like the traits that are like shared among the things, like on the actual, what is it, cladogram?
Yeah, like these are mutations. That's all they are. They're DNA mutations, and it tells you at what point in the cladogram those mutations happen. This is a cladogram, but instead of derived characters written out, it tells you the actual mutation that led to the derived character. Right? That's the first thing. They show you when the mutations happen. What's the second thing? It really helps.
All the mutations are highlighted.
They color code them. Yeah, it's really nice. So yeah, Vireo, we need to find out who's most closely related to Vireo. That's easy because there's a mutation there. The other organism that has it is going to be the one that goes there. 31C to T. It tells you of nucleotides, right? 31, we go to 31. C to T means the mutation turned a C into a T. Well, there's Vireo. The other one that has it is Diphyllodes or Diphyllodes. I don't know how you say it. I don't even know what these are. Birds? They're birds. Okay. So Vireo and Diphyllodes go together. Okay, the other one on the fork with them should have the first position, a T to a C. So here's Vireo as a C. Here's Diphyllodes. The other one that has a C, Manicoidia. So Manicoidia goes there. And so now, well, why even worry about the rest of it? We know, right? We know that there's only one left and it's got to go in the last spot. So let's do that. Okay? Questions about that one? We are going to skip this one because everybody got it right. Just another matching up the bars. I guess I should answer it. That was 2. Number 5. How could genetic engineering be used to produce a more successful crop in a hot, dry climate? This is a terrible question, which leads us to, I don't know if I've mentioned this yet, the questions they release, that the state releases from old EOC tests. Do you know why they release them?
People got them.
Sometimes it's because too many people got them wrong. Sometimes it's because too many people got them right. Sometimes it's just because it was a really lousy question. It's because they threw those questions out. And so something to know about end of course exams, first of all, is that your practice tests are often made up of the questions that didn't cut it in the real test. So if some of them seem weird or unfair or too easy or too hard, that's why. Okay? So how could genetic engineering be used to produce a more successful crop in a hot, dry climate? A, non-essential DNA can be removed from the genomes of the crop plants. Does that help anything? I don't think so. B, DNA from plants adapted to dry areas could be added to the genomes of the crop plants. Does that sound right? Maybe. It's not specific enough for me. I mean, shouldn't it be DNA that we know helps them be adapted? But I guess it sort of sounds right. Let's look at the other ones. C, RNA from a variety of non-agricultural plants could be added to the genomes of the crop plants. Would RNA do anything? I don't think so. That seems weird. mRNA transcripts, indeed, from dry weather crops could be added to the fertilizer used on the crop plants. Does adding anything to the fertilizer genetically, is that going to go anywhere? No. So you're right. B is correct. It's just not a very good answer, but it is the least wrong of those four, so that is what you should pick. All right. This thick 6 you guys nailed. I was super happy that all of you remembered that you can't use, if they give you the DNA and they're asking for you to use codons to find amino acids, you got to transcribe it yourself. You got to get from GGG to CCC to know that it codes for proline. And all of you did that correctly, I think. So we're going to skip that one too. It was proline is unchanged and asparagine is unchanged. No, it was, yeah. Yeah, because it didn't cause any change because it was in that wobble-based spot. Number 7. Growth curve for a population of Ohio gray squirrels, no predators and no limit on resources. That's called what kind of growth? Exponential. In reality, natural environments will always have resource limitations. Which graph realistically predicts growth for a natural squirrel population with no predators? I don't know why they saw fit to say no predators, because that kind of makes you think it's going to keep being exponential. But the point is, there's always resource limitations. So what are we looking for? Well, this has everyone dying. And this has everyone dying. And this one is just the same as the first graph. And you know that realistic logistic growth looks like that. So that's the one you should pick. Remember, it bounces around its carrying capacity as it overshoots. Number 8. Scientists study anaerobic fermentation in yeast cells incubated at different temperatures. The amount of CO2 detected at each temperature in 2 trials is shown in the table. Why would they measure the amount of CO2? Why is that useful? What does that tell them? What's carbon dioxide in fermentation? Why are those two linked? This is the oxygen debt. Oh, we're not worried about oxygen debt if we're just looking at yeasts.
Because like during like the Krebs cycle, the CO2 is like shot off with the when you go through the.
Not the Krebs cycle. Krebs cycle shut down when there's no oxygen. But remember alcoholic fermentation? Remember how yeast take the product of glycolysis, which are pyruvic acid and NAD+, and convert it back into NADH, and they convert the NADH into NAD+, and in the process, alcoholic fermentation turns the pyruvate into CO2 and alcohol. Does that ring a bell? It's like what makes beer, it's like what makes bread rise, the CO2 part. So the first question here is, what are fermentation products? And so hopefully you knew to choose ethanol, which is the alcohol part, and CO2, which is where the carbons go, right? Or where the third carbon goes. And so the reason they're measuring mass of CO2 is because CO2 is a product of fermentation. More CO2 means there's more fermentation happening. So optimal fermentation temperature is what? Why? Because that's the temperature at which we saw the most CO2. Exactly. All right, next. In a particular species of mice, ooh, this is a nasty one. A single gene determines tail length. This is where that habit of writing things down initially comes in handy. Oh, never mind. Big B is short tail. Actually, X big B is short. X little b is long tail. Shading indicates short tail, which means this male is what genotype? He's got a short tail. Remember, males show you everything they've got. He's X, Y, and what's on his X? Big B or little b? Big B. She's got a long tail, which means on her two X's, she has what? Two little b's. This is easy once you get it, because this is supposed to be a female. Individual 6 is female. What's her genotype? Well, her genotype is going to be, well, what came from dad, which is X big B, and what came from mom, which had to be X little b. She is a carrier. She is X big B, X little b, which is letter A. Moving on to 10. A student is asked to draw 4 cells. Which drawing is a representation of a prokaryotic cell? I don't love this one, but we can at least knock things out right away. What do you know? How did you do this? How did you deal with this? Eli, what did you do? So what one can you knock out right away? What one do you know isn't a prokaryote? B, because that looks like an animal cell to me. There's a clear nucleus in the middle. There's mitochondria. We know prokaryotes don't have either of those, so it ain't B. What else is obviously not a prokaryote? D is a plant cell. You've seen plant cells. That's not a prokaryote either. So you're down to A and C. And I don't love this one because A sure looks like, I think they mean A to be a protist, like an amoeba or something, and that's a little nucleus, but it doesn't look very good. But it's got these little bubbles. I guess you can call those organelles. This just has DNA floating around in front of God and everybody. No nucleus. It's got to be C. All right. Is the last one? Yeah, last one. Some bacteria have developed a resistance to antibiotics through random mutations. One such mutation allows bacteria to produce enzymes that inactivate antibiotics when they enter the bacteria. Which statement describes how this mutation occurs? And what the question is basically asking is, how do mutations happen? Which you know. How do mutations happen? We learned 2 ways they happen. What were they? Yeah, we learned those types of mutations. But what causes them? 2 things. Oh, somebody knows. Number 1, we said mistakes. Mistakes in what? Danny? Mistakes in doing what cause mutation? DNA replication. The other one was mutagens, right? Chemicals and radiation. So letter A, the mutation occurs when nucleic acids are replicated. That sounds right, doesn't it? We just said replication. Nucleic acids are DNA and RNA. That's it. You can read the rest of them. They have to do with chloroplasts and mitochondria and moving across the cell membrane, none of which has to do with mutations. So that was part 1 of the practice test, which, by the way, the averages were much higher on this than on part 2. So let's look at part 2 now. All right. We're going to skip 3 and 8 on this. Let's see why these were so hard. Ooh, this one. Diagram shows an incomplete cross-section of a cell membrane. You should recognize the phospholipids. 3 different particles across the membrane. This is basically like deeper problem stuff. Plus, when we talked about active transport and facilitated diffusion type stuff. 3 molecules are oxygen, glucose, and sodium. And the 3 things embedded in the membrane that you can choose from are channel, which you should remember is channel proteins, right? Phospholipids, which is just what the cell membrane is made of anyway, and ATP pump, which you can think of as the sodium-potassium pump, okay? So, first of all, what's the main rule of diffusion? Stuff goes from what to what concentration? High to low. And that is happening here, high to low. Here, high to low, but not here. This one's going low to high. So what does that one have to be? What's the only thing that can go from low to high concentration? Nicholas. Nobody's answering. I'm just going to start picking on people. A pump. Yeah, the sodium potassium pump. Active transport is the only thing that can make stuff go from low to high concentration. So that has to go there. Hopefully you remember that oxygen is the one that we had in bigger problems that can always get across the cell membrane, along with carbon dioxide, without any kind of special channel or pump. And so the one that has oxygen, which is the green dots, that's going to be the one that's just the normal stuff the membrane's made of, right? Phospholipids. And then glucose, if you remember that glucose channels are a thing, and that facilitated diffusion is when you allow a big molecule like glucose to just diffuse through a specially sized hole, that would be channel going there. Any questions about that? All right. A biologist conducting a long-term field study noticed that there were 2 types of mice. One had webbed toes and one lacked webbed toes. A dam was built and flooded it into a wetland. Over the years, all mice living in the valley were found to have webbed toes. None were found with non-webbed toes. What statement best explains the observed change in the mouse population? Right now, they are basically probing you for a misconception. That's what this question is. They're seeing if you have the misconception that hopefully I've been idiotic enough in my exaggeration of that you remember that you can't just go, oh, it's cold out and grow hair. Doesn't work like that. Individuals do not evolve. So that means that letter B, the mice grew webbing to move more efficiently in the new environment that a lot of kids are going to pick. You know better. You know not to pick that because the mice can't grow webbed toes, at least not in a way that's going to get past them. Webbed-toed mice survived better and reproduced more than mice without webbed toes. I try to phrase everything I say about evolution and natural selection in that way. It's not that they got webbed toes. It's that the ones that already had them survived and had more babies. Good? So that one is C. Number 3, everybody nailed, which I was impressed with. That's not the easiest thing, this cladogram. But yeah, you did it, so good job. Number 4. Which pair of organelles works together to give structure and support in animal cells? This is kind of tricky just because there's something correct in every answer, right? There's cell wall in letter A, but cytoplasm is too general. That's not, that's everything and stuff. Cytoskeleton and Golgi complex, cytoskeleton's right, Golgi doesn't support. Cell membrane and cytoskeleton, that's the one, right? Cell membrane is the outer part of the cell. Cytoskeleton is all through it, providing structure. It's letter C. In most species, scientists have observed that related individuals share about 80% of the same genes. Cheetahs, however, are known to share 99% of the same genes. What does this lack of genetic diversity imply about the history of the cheetah population? If you don't know already, they like to use cheetahs because if you don't know already, cheetahs almost went extinct at a certain point. They're still endangered, but they've repopulated somewhat. But what happens when you're down to like 10 individuals on the whole planet, and then they repopulate, is that the new population looks way more like those 10 individuals than like any cheetah that came before. It's almost like a version of the founder effect, where a small population found the new population, except it's called a genetic bottleneck. When you almost go extinct and then you repopulate, you're not going to have much diversity for a while. Okay, so that's the idea. This is one of those questions. Cheetahs never have experienced mutations. You know that's wrong. Letter C, cheetahs interbred with other large cat species. That would create more diversity. That's not it. Cheetahs share a common ancestor with other large cat species. Well, yeah, that's true, but it doesn't matter to this question. B, cheetahs have experienced mutations, reproductive isolation. What? That's not a sentence that makes sense. But the other ones are so obviously wrong that if you run into a question like this, where you're like, I know this is a typo, they have to throw this question out. You can at least get it right. Right? You can get letter B. Number 6. 4 different cell specimens recording information about each cell. Which cell specimen is a prokaryote? All you care about is one word. What is it? What's the word you are looking for? Ella? That Ella. What's the word you're looking for in this to know if something's a prokaryote or not? Nucleus. Yeah. Here's that word. DNA in nucleus. Yes, yes, no, yes. It's the no. Right? The one without a nucleus, that's the one that's a prokaryote. So number 3, letter C. Number 6? No, 7. I'm supposed to, no, I skip it. All right, diagram shows a plant cell with several structures numbered. Identify 2 cell structures involved in the function described. Modify proteins and transport them out of the cell. How'd you do? This is hard because they don't give you the names. If you're a name person, you didn't have that. You had to go by the diagrams, and you had to remember that they often draw the structure we're looking for first, the one that modifies proteins. Which one is that again? ER, yeah, they often draw that grouped around the nucleus. So that's number 6. So number 6 is one. And then the one that ships stuff out, the post office of the cell, the shipping department of the cell. Ethan, which one's that? Golgi. Do you tell which one's the Golgi on there? So it's not the green thing, obviously, and it's not the thing that looks like mitochondria because that's mitochondria. It's not the big old vacuole. It's this one right here. It's not a great drawing, but when you see that flattened stack of membranes, that's how the Golgi is almost always depicted. So number 2 is the other answer. All right, next. Is this 8? Everybody got, you guys are really good with the cladograms, so that's great. Get that one. Stone crabs, 3 phenotypes: dark brown, big B, medium brown, big B, light brown, little b. You probably already figured out that they're not doing it the same way we do it. When we do incomplete dominance, we use all capital letters and one with the mark, right? They use big and little, but they tell you it's incomplete dominance. They tell you that the predator favors medium crabs, which would be big B, little b's, while avoiding dark brown and light brown crabs. This is kind of a version of disruptive selection, if you don't remember that. So predict how this selection pressure will affect the allele frequencies. This is an interesting one. I like this question. Tell me how you reasoned through it. Tell me how you got it right, Logan.
I get it right.
You didn't get it right? Who got it right? Raise your hand. Oh, those are just the ones who are brave enough. Way more than 3 of you got it right. But Nicholas, go ahead.
So if the medium brown crabs are gone, that would be the dark brown and light brown, and the dominant and recessive are still left, so they still exist.
So every time a predator takes a medium crab, is it affecting the allele frequencies at all? No, because it's taking one of each. There's only 2 alleles. if you take one of each kind every time, they stay the same, don't they? Yeah, so the answer is no change for both, which I think probably threw a lot of people, which is why they threw this out, but you would get it right, I hope. Is this the last one? Last one, rabbits in Australia, 1800s, overpopulated, few natural predators, because there've never been rabbits there before. To control it, they introduced a rabbit-specific virus. What could go wrong? And their numbers greatly decreased. However, after several generations, the rabbit population began to increase again. Which statement explains the new increase in rabbits? A, the rabbits interbred with native rabbit species. Isn't the whole thing that there weren't any rabbits? Yeah, that's not right. B, some of the rabbits had a natural immunity to the virus. Does that sound right? That sounds like how we talk about natural selection, right? It's not that they grew an immunity, but some of them already had an immunity, and they're the ones that repopulated when the rest of the rabbits died. C, some of the rabbits learned to survive even though they were sick. No, that's not evolution talk. And D, rabbits were able to have more offspring by changing their reproductive cycles. Again, that's just change during your life. Doesn't work that way. It's got to be B. So that is just kind of my attempt to show you how to think through these questions. I hope it helped. Do you have any questions? I think I'm going to save the final pep talk for Tuesday. How much time we got? 35? Yeah, so why don't you take 10 minutes, work on these sheets. Okay, I promise these seem like just stupid matching sections, but I promise thinking about these and getting these right will help you, because this is all the stuff they think is important. That is a view inside the heads of the test designers. Amanda. I will show you.